Hi everyone, so today we're going to talk about undergraduate research of the antibiotic crisis. I'm Kendall.
Hi, I'm Alex.
Okay, so we're going to first talk about what the antibiotic crisis is and what antibiotics are. So most antibiotics come from microbes. Antibiotics are a medicine that prevents infections caused by bacteria. Antibiotics kill bacteria or keep it from reproducing or copying itself. We use antibiotics in medicine and surgery, and it has extended the life expectancy. But there has now been an issue that has arose against antibiotics. It has become a major concern to the global health worldwide. So why? The answer to that is the bacterial resistance that is occurring towards antibiotics. It can affect many individuals regardless of their age or region. So what causes this bacterial resistance to antibiotics? Well, there's a few. So first I'm going to talk about mutations. So when bacteria multiply, errors occur, creating mutations, which then causes a change in the nucleotide sequence of genes. Another most well-known reason is that antibiotics are being overused every day. Antibiotics being overused has caused evolution to occur and resistance to the antibiotics. Another factor contributing to this resistance is that antibiotics are being prescribed inappropriately. They are also being widely used in the agricultural industry for supplements in livestock. It is said that 80% of our antibiotics are used for livestock and becomes a problem because that is our source of food. So if the animals are taking the antibiotics, it is then transferred into humans after consuming the food. Due to economic and regulatory difficulties, newly developed antibiotics have not been able to be produced, which has caused a major concern to microbiologists and infection disease specialists. So why is this topic important? Well, antibiotic resistance have been spreading across the globe. Infections that are antibiotic resistance have now become a burden to health industry and has become a costly issue. So resistance to antibiotics will be an overall threat to the global population. So now we're going to shift on what we did for our experiment. And to the right is a little flow chart of what we did. So I'm going to first talk about week 1. The soil was collected for this experiment at California University of Pennsylvania behind Frick by Dr. Valkanis. To do this, we then did serial dilutions with 1 gram of this soil and then transferred into 9 milliliters of solution, making 10 milliliters altogether. And then that was transferred until a 1 to 10 dilution was prepared. And 5 of those 1 to 10 dilutions were performed by Dr. Valkanis, where she then plated the last 4 dilutions to obtain single colonies. The plates were then incubated at 30 degrees Celsius for 3 days. We needed to isolate the colonies due to the dilution plates mixing over time. So we used the pick and patch method, which involves picking an individual bacterium from a colony. The bacteria were then picked from the dilution plates using a toothpick and then patched on the new agar plate, which was then called our master plate by Dr. Valkanis.
So from week 3, we use selective and differential medias to help us learn more about the bacteria. And from those, we use the McConkey agar, which selects for gram-negatives. On it, pink colonies and red media are indicative. White colonies and yellow media are indicative of gram-negative and non-lactose fermenting bacteria, and no growth is indicative of gram-positive bacteria. On the other plate, the mannitol salt agar, opaque and pink media is indicative of gram-negative and non-mannitol fermenting bacteria, and growth in yellow media was indicative of gram-positive mannitol fermenting bacteria. No growth was indicative of the gram-negative. In week 4, we did patch plating using the escape pathogens, in which we used Pseudomonas aeruginosa, Staphylococcus epidermis, and Bacillus aureus. From there, we coated 3 plates in the bacteria, and then we took samples from our master plate and put them on to analyze the colonies. What we found was that the CFU number for 10 to the 4th colony was 1.96 times 10 to the 6th. The colony morphology was also analyzed, observing things such as texture, size, color, if it was raised or if it was flat, and most of the isolates shared characteristics from the master plate. So our results demonstrated in the differential and selective medias where in the MSA plates, most of the isolates were opaque with pink media, indicative that they were gram-negative and non-mannitol fermenters. In isolates 1, 2, and 12, there was no growth. Only in isolate 9 was the gram-positive bacteria that was mannitol fermenting, as concluded with the yellow media. In the MAC plate, there was no growth for 1, 7, 9, and 10 through 12. 6 was positive for being a lactose fermenter, and the rest were weakly positive. Antibiotic screening tests were then carried out with the Pseudomonas aeruginosa, Staphylococcus epiderma, and the Bacillus aureus. In the 3 tables of the antibiotic screening, we had no positives for antibiotic activity. Everything was negative. The total prevalence of our individual data was 0%. However, other students in our class were able to get positive results, and from there on, they're going to go on and further analyze their bacteria. In conclusion, we did not have any antibiotic producers. However, undergraduate students are able to contribute to finding new antibiotics through the Tiny Earth Project, which is a great way to mass examine soil, other samples such as water, and trying to find antibiotic producers.
And to add on to what Alex said, this is an important topic because we use antibiotics every day, and it is going to be a major concern to the public health if this resistance keeps occurring. So we said if we were to further extend this experiment, we would possibly test them against eukaryotic organisms to see if they are maybe harmful to them. That's all we have for today. Thank you.
Yes, thank you. And we would like to thank our Professor Dr. Balkanas for getting us involved in such important and significant research, especially in the field of microbiology and the healthcare sciences. This was great and meaningful, and we were happy to be a part of it. So thank you.